Yep. Ugh.
Hi, Jess. Hi, Liz. Hi, Madison. Sarah. Tony. Jess. Alexa. Hey, Jake. How are you?
Good, you?
I'm doing okay, thanks. Okay, we've got 12:30. Why don't we get started? Good afternoon, S&P. Happy anniversary. I was saying to my earlier class, this is our 1-month anniversary. It's hard to believe we've been going for a month, but we have. And so far, so good. So thank you for a good 1 month. And I've got some really great news. As you can see, we've got a paper coming up due right here on Wednesday, right? So we're almost there. And this will be the opportunity for us to have another in-class discussion. So we're going to have the chairs in a U, and you will have all written about 1,000 words or so on that topic, connecting different ideas together, and we'll have you converse during that time. Hopefully I'll say go, and the conversation won't stop thereafter, okay? You don't need to prepare any videos. You could have a video if you wanted to, but you're really going to be talking about your papers and the ideas therein. So looking forward to a good discussion, and then after that we'll be doing color perception. We'll continue into different kinds of features of visual perception after we've laid the groundwork in understanding the eye, and today we'll get to the eye and brain, okay? All right. Well, we got clouded out last night. I was really excited this time yesterday, thinking that, wow, we'd get to see something that hasn't been visible at this location, this latitude, since 1982, I believe, and we got clouded out. So we only have to wait until 2029 for that to come up again. Just 14 short years, and we'll get another crack at it, assuming we don't have cloudy skies in 2029. But anyway, it would have been cool if we had done that, because we would have seen the full moon, and would have been able to put the full moon into our blind spots. So, how big is our blind spot? Somebody help us out with that? Yeah, thanks.
The width of our thumbnail from an arm's length away.
Okay, so we can, it's the width of our thumbnail from arm's length away. It's one way to think about that, okay? What else can we say? Actually, that's not the full width of the blind spot, but that's 1 degree of visual angle, okay? But that's okay, because that's how we express the measurement of how big the blind spot is. So how big is the blind spot using that measure? Can you go ahead, Liz?
Oh, I don't know if this is the answer you're looking for, but I was going to say something about the hole in the back between our eyes that allows us to have the blind spot to build up a hole in our vision.
Okay, there's a optic disc there. Yeah. Okay, there's the optic disc. All right. We're trying to figure out how big that is. By the way, what is the optic disc made out of? Why do we have an optic disc? It seems to be giving us a blind spot. What kind of crazy design was that? Yeah.
Axons of the ganglion cells.
Okay, the axons of the ganglion cells, right? And they're trying to transmit information from the retina to the visual cortex, right? Okay. So we're still on the hunt. We now know that what the ganglion cells are doing. They're forming that optic disc. We know that it's about, as Claire correctly said, it's about the width of 1 thumbnail extended at arm's length. Okay. So how big is that thing? Did you have that? Maybe Claudio's got it. Okay.
It looks like 12 full moons.
12 full moons. Okay.
So 6 degrees.
6 degrees. Okay. How many micrometers? Degrees is an angular measurement, and that can be helpful when we're trying to describe how big something is on our nervous system, right? Because it might be the case that I'm writing up a journal article and I'm trying to tell my next reader or the next person who's going to run this experiment how to reproduce this study, and need to specify how the central nervous system was stimulated. So it's very helpful to be able to specify that. But sometimes we want to specify it in angles, degrees of visual angle. Sometimes we can also specify it in how large it is on the retina in a linear measurement, like in millimeters, but we don't use millimeters, we use micrometers. Okay, does anybody remember how many micrometers that was? Okay, so how many degrees of visual angle? It's 12 full moons. How many degrees of visual angle is that? 6 degrees, okay, 6 degrees, okay. And a given degree of visual angle occupies 300 micrometers on the retina. Okay, 300 micrometers. That's 300 millionths of a meter. Okay? So if we have 300 of those micrometers in 1 degree of visual angle, and we have 6 degrees of visual angle for our optic disc, what's that work out to be? How many micrometers? 1,800. Yeah, 1,800 micrometers is what we've got. Okay, so we could have had the full moon, even the super full moon probably would have fit in 12 times, maybe a little less than 12 if it's super full moon, but you get the picture. So we'll find out in 2029 how we do on that. Okay, so we got into that conversation because we were speaking last week about visual physiology. We spent a lot of time on the eye, and we tried to figure out in part where the eye comes from. And we concluded last week by having a couple of chapters by Richard Dawkins, who is an evolutionary biologist and a zoologist, and he wrote Explaining the Improbable, and we had a good conversation about that. He also wrote that important chapter from The Greatest Show on Earth, and that was the title of my email to you all. I thought we were going to have a great show on Earth yesterday, but we got clouded out. And we talked about the Lenski experiments from Michigan State. We got a really good description. We didn't go all the way through, though. Just to remind us, can somebody tell us a little bit about the gist of the Lenski experiments? How are those organized? What were some of the ideas about the Lenski experiments and the procedure? Okay, thanks, Alex.
Well, one of the ideas, I think overall, was that there were different evolutionary routes to the same conclusion, and that the odds of that happening were astronomically high when they happened.
Okay.
It was not by chance.
Okay, right. So it is certainly the case that when we think of something like the eye and we ask about how complex it is and how could that have come about through evolution, well, we can actually set up an experiment and we can have, as Gabby correctly explained the other day, and Alex is alluding to now, we can have 12 lines that might contain something that is going to be mutating and varying over time. It might be flasks filled with different kinds of bacteria. And there are different names that we can have for these different types, and we can basically take a new generation every day, right, for something like tens of thousands of generations, right, tens of thousands of days. It's probably going to take a huge amount of space, but you can do that. And what's really cool is if at some point you find that these bacteria are behaving in an unusual way, you can basically dial back into the evolutionary sequence because they freeze the sample every single day for I don't know how many generations, 1 generation per day for tens of thousands of generations. And you can say, let's go back a couple of hundred generations, and let's unfreeze that. We'll take a sample, we'll refreeze it, and we'll take our thawed sample, and we'll take a look at what's going on there in that genetic pool, and we'll see if we've observed that mutation just yet. And we can even now multiply that one. We can make 12 flasks out of that, and we can now run that experiment 12 times. So you basically have evolution in an experimental jar, right? You can just recreate, you can let evolution recreate what it's created anew each time. And maybe it comes up with slight variance on that, but it can converge onto a given solution time and time again. So really, really elegant. Has to be done very diligently, but a very powerful explanatory mechanism. So who remembers reading about those experiments? Does that sound about right? Like we've got a pretty good description of the Lenski experiments. Okay, what I wanted to do next was get into the one question that we didn't get to on Friday. And this was still on the Lenski experiments. I think the folks on this side of the room may have been dealing with this question. And we were trying to briefly summarize how the research that is described in that chapter on the Lenski experiments addresses the issue of so-called irreducible complexity. So before we pick up that question, let's just unpack that phrase. A lot of us who study evolution are aware of the concept of irreducible complexity. How many people had heard of that before? The idea of irreducible complexity. Our biologist has, okay? How many people had not heard of that before? Irreducible complexity, okay? Could I put you on, could you help us out with, maybe not even the experiments just yet, but just what is the idea about irreducible complexity?
Well, I think it's just the idea that that we could change from one organism to the next is so great that it'd be difficult to occur through natural selection.
Okay, very good. Right? So the change from one organism to the next might be so great that we can't get there by random mutation alone, right? So the idea that Darwin was putting forth, even though Darwin didn't have what we now call Mendelian genetics, right? He wasn't aware of that at the time. But in his observations, when he was about your age, and he was traveling to the Galapagos at about the time this college was founded, he sees these subtle variations in various life forms. He's trying to explain the subtlety of descent with modification. Here's a generation, their offspring will look pretty much like them, but not exactly like them. So we get one small change at a time, very much like Lenski's experiments, right? So then how do you explain what seem to be relatively large changes, right? I mean, how can you go from something like a simple bacterium or a single-cell organism to a human? And not even that far. How can you go from a single-cell bacterium to a human eye? Right? I mean, that's a lot. Right? We learned about the complexity of the eye. Somebody, it might have been Abby, who told us about the definitions of complexity. And one of the ideas there is that one of the criteria for something being complex is it has to be heterogeneous, right? So the eye, unlike pudding, is very different if you cut it this way versus if you cut it that way. Pudding is the same any way you cut it, right? But the eye is very, very different. We have this remarkably complicated organism. How can you get there in these little tiny steps? Will those ever be enough to add up to something like the eye, let alone a full-blown human? Might it be the case that if we work with hierarchical reductionism, which we said was reducing things step by step, at some point you get to a level where you just can't bring it down any further. You can't go any lower than that, because if you would, the organism would stop working, or the eye would stop working. You can't reduce beyond a certain level of complexity. And this is often a claim that is made by people who don't buy into evolution. They'll say, well, I found a structure in nature that can't be reduced any further, because if it were reduced any further, it would become dysfunctional, and then it would have no evolutionary survival value. One way to think about this was a very famous question that was posed to Charles Darwin. When people were hearing about his theory, and they began to think about it, okay, why don't we try to play the clock backwards? Maybe I'm a little bit like my parents, or a little bit like my grandparents. We play it all the way. But sometimes you get to, simpler versions that don't seem to do anything for the organism. And here was the famous question, asked rhetorically. Mr. Darwin, what good is half a wing? Right? What good is half a wing? So you see this animal with these beautiful wings, and they're flying very beautifully. Well, the parents before that would have had very similar wings. The parent before that would have had similar wings, but they might have been slightly shorter. What good is half a wing? And what can you do? But at some point, the wing length must have been roughly half, if we believe in evolution, that we have this step-by-step random mutation that we see so elegantly in the Lenski experiments. So Mr. Darwin, what good is half a wing? That doesn't buy us anything. Right? That's not functional by itself. Who's following the problem? Okay? So it's almost as if we have these structures, or we might believe that we have these structures that are irreducibly complex. You can't get from this level to this level with a simple random mutation. You need something seemingly bigger than a random mutation to get you up to, from a half wing, which buys you nothing, up to a full wing, which allows you to fly. And flying very clearly has evolutionary advantages. By the way, I wonder if we can interrupt ourselves there. What good is half a wing? If I can't fly with half a wing, what good is half a wing? Yeah.
I guess from still parent, I guess being able to rotate on a dime while running at high speed would be important.
Okay, all right, so maybe I can't fly, but maybe I can turn faster than Meg can. She and I are trying to run away from the bad guy evolutionarily, right? And if I can put my half wing into the right position, and she has like a quarter wing, and I can get just a little bit of lift, and that makes me turn the corner faster than she does, I have a slight evolutionary advantage. So maybe it wasn't there initially for flying, it bought the organism maybe something else. Okay? Can somebody think of another example? What else might half a wing do for you? Okay, next.
Or like attracting a mate, so it could just be like a feature that was...
Okay, it might have been there not for natural selection, which is how do you stave off death, but it might have been there for sexual selection. How do you attract a mate? Okay, so maybe it served a different kind of a function. And then longer wings got better and were more sexually attractive, and then all of a sudden these wings start, if you will, literally taking off. Right? Okay, so now we can run a little bit, and we can fly a little bit. We can fly from me to Claudio, and that might help out in a jam. Another example that's sometimes given is if you're imagine that every once in a while these animals are running to get away and they have to jump off ledges occasionally. Well, if you have half a wing, it's kind of like having half a parachute. It's not that great, but it's better than no parachute, right? If it affords some resistance, maybe you land slightly more gently, and you might break a limb, but you don't die in the crash, okay? So half a wing might buy you something, right? So maybe there is some evolutionary advantage to having something that's not yet fully formed, right? Okay? It's one of the ways of thinking about it. But it does seem that every once in a while we're going to take more than just a small step. We're going to take a really, really big step. And this is partly what gets us back to this question number 2. There were a couple of really, really big steps in Lenski's experiment that might begin to address the issue of irreducible complexity. This notion that, sometimes we get a relatively large leap, seemingly, in evolutionary development. And it's not entirely clear why that would happen all of a sudden. Of course, some people would believe that the reason you see a large leap in evolutionary development is there's, if you will, the hand of God. God intervenes, gives evolution a little push, and all of a sudden we go from here to here, not by this, which is a purely natural function, but because of some kind of supernatural intervention. That would be the claim. But Lenski seems to have a different way of getting there, okay, in Lenski's experiment, right? Using only this. And this was described on those pages in that chapter. Somebody help us out with what he observed that seems to have given us something like a big step. Okay, thanks, Kim.
So I think it was the addition of multiple mutations added up to the ability to use the other food source.
Okay. Yeah, that's exactly what we're looking for, right.
So he said you couldn't have the ability to digest citrate unless you had a bunch of different combinations already suited.
Yeah, very, very interesting. So you might remember that they had taken a little drop, I'll call it, or a splash of glucose, and that was what was being fed to the bacteria. And the bacteria, over time, would grow something like this. Let me draw a simple graph where we have the number of generations on the x-axis. And of course, he's got tens of thousands of generations, so number of generations. Okay? And he has something like the population size over here. Okay? And as the organisms are mutating, they get better and better at making best use of that little splash of glucose. And so it goes something like this. And it tends to fall off, and it does grow, but it grows pretty modestly. Step by step, we get something of a modest growth. And then all of a sudden, at generation, whatever it was, something like 33,000 or whatever, all of a sudden, ding, it goes like that. Right? How do you explain that? Right? Now, some creationists might say, well, that's the hand of God right there, right? Because you can't just have that kind of a takeoff with random mutation alone. Right? But as Gabby's correctly pointing out, what happened was there happened to be a particular sequence of mutations where all of a sudden now, instead of just being able to eat and live off of glucose, you can make use of the citrate that was also in there. So because they happen to have a particular pattern of mutations, and none of the other tribes did, that one tribe that happened to evolutionarily stumble upon this mutation in combination with this mutation, and in that order, all of a sudden, that tribe and that tribe only can now metabolize citrate. And that allows a larger fraction of the population to grow, so they get this big spike, and it stays there. And none of the other tribes do that. Right? They talked about mutation A and mutation B. A lot of the tribes had developed mutation A, but not B. Others had B, but not A. And I think there might even have been something in there about some of them had A first and then B second, and that didn't work. It had to be B first and A second. There was something about that sequence that allowed the biochemistry to do a lock and key and unlock the capacity, essentially, to digest or metabolize citrate, and that bought it. So all of a sudden now we get this huge hand of God effect. But all we really needed was this: random mutation and non-random selection. Right? Nothing unnatural was happening in those tubes. And what's really cool is you can dial back and say, all right, so let's go 2 generations, 2 days before that mutation appeared. And you can let those run for a while. You can go 2 days after it appeared. You can let those run for a while. He's got the whole ball game in his set of test tubes. I think it's really, really cool. Who's following that? Right? So you can imagine now, over many, many, many days, most days are going to look like this, but every once in a very long while, it was tens of thousands of generations, you get a spike. And you can imagine it's going to stay there for a while, right? who knows? After another 10,000 years, there might be another spike. So generally, it's a slow process, and every once in a while, it's punctuated by a steep spike. Okay? And maybe a process like that is how we get all the way up to something as complex as the eye. Okay, enough on that. Unless there are other questions about the Lenski experiments from Michigan State University. Okay, I wanted to share with you a brief series of PowerPoints that I think are really, really interesting, that you've not been exposed to before, but they kind of develop this idea. Okay, they're not quite as elegant as the Lenski experiment. I was showing these slides before the Lenski experiments were published and before Dawkins so elegantly wrote about them. But I did want to get to just an idea about generally how you can show in a test tube or in some other kind of controlled environment the replicability of evolution. So we can ask about different kinds of scientific criteria. We say that science typically has to be falsifiable, right? We're putting forth falsifiable hypotheses. We want things to be parsimonious. We want to have nice, clean explanations. We want to be able to replicate things. One challenge with evolution is that evolution began many billions of years ago, and we can't just go back and recreate billions of years of evolution. So how can you begin on a replication? You can embark on what's called a conceptual replication. You can say, well, these are the key components of evolution, and we want to demonstrate them, as Lenski has done in his experiments. Another simple example is something like the mosquito longevity study, and there are variations on this that people might be familiar with. The idea is that I might have an aquarium where I put a lot of mosquitoes, and the mosquitoes are living to a certain age. Okay? Here's the older mosquitoes, and they have a higher age. Here are the younger mosquitoes. And in this distribution, we have a bunch of middle-aged mosquitoes, a few elders, and a few young ones here is what we've got. Right? So that's the idea. And mosquitoes will reproduce at particular temperatures. Okay? So what we can do is we can cool our aquarium, or we can heat it up such that it's at a temperature that affords reproduction or that doesn't afford reproduction. Okay, so there might be some kind of a temperature threshold here, and maybe our actual temperature is above threshold, so the animals can now reproduce in here. But what we can do over time is we can create this kind of a pattern. We can say that we're going to cool the aquarium for some period of time while an animal is young, okay? And then we gradually let it warm up, and at about the halfway point, we'll now pass the temperature that is needed for reproduction. Okay? So if we tie that back to what's going on over here, a lot of these animals are going to die before it gets warm enough for them to reproduce. Who's following that? Okay? But the animals that have slightly greater genetic longevity are going to live long enough where they can be in an environment, an aquarium, that is warm enough where they can reproduce. So now the only animals that are essentially allowed to reproduce are selected non-randomly from this population. This side can reproduce, this side can't, right, because they've died off. Who's following that? Okay. So if you do that, and then you play this ball game over and over again, and you keep on just pushing that crossover point a little bit further to the right, as if this were a psychometric function, further over to the right, further over to the right, you can actually increase the longevity of your mosquito population. Right? So a similar kind of idea that you might have for breeding animals. Maybe you want a particular breed of dog to have a particular kind of a trait. Same kind of an idea here. We can show evolution in action using this. Right? So we can select for a particular kind of a trait. Okay? So, and then you can go and push this to all kinds of extremes over many, many generations. Okay? And this doesn't necessarily prove that evolution was working 3 billion years ago, but it is what we might call a proof of principle, right? That if we have this random mutation, as shown by these different ages that we have randomly mutating across the age spectrum, we're going to non-randomly select them. We can now push a particular species to have a particular trait. Okay? Now, of course, evolution isn't driving toward any particular goal in mind, right? Evolution is the blind watchmaker, right? It works with what is at hand, not what is in mind, but what it has in mind. Okay? But it's still enough to use those two simple steps to get all kinds of descent with modification. Okay? And over billions of years, amazing things can happen. Okay? Who's following all that? This is just another example, maybe, of what Lenski's got going on there. I did this the very first year that I taught this course, and then one of my students said, well, I think I agree with all of that, right? But how do you get life going in the first place? Right? I mean, how do you get the first self-replicating organisms going in the first place. Once you get that far, then I can see if you have a bunch of self-replicating organisms, I can see how we can play the evolutionary game and get them to mutate into this, and into this, and into that, and every once in a while you get a spike, right? But how do you get the whole ball game going in the first place? So, which I thought was a great question. Does anybody have some ideas about that? How life got started, presumably in the first place, so that we could eventually wind up with a human eye, so we can have an S&P class. Natural, biologically based ideas for how life got started in the first place. I don't know if our biology department spends time on the origin of life. So Darwin is about the diversity of life, right? On the origin of species, how does one species come from the next? Okay? It's a theory about the diversity of life, not about the origin of life. Okay? So here's one of the ways of thinking about this. You can see that as we use evolution, we can move from here to here on a number line, here to here on a number line. If you can imagine a number line starting at 0, how do you get from 0 to 1? At some point there was no life on Earth, and then all of a sudden there's life on Earth. How do you make that transition? How do you go from 0 to 1? It's a big qualitative step. Okay? Well, here's an answer that actually doesn't work, but set the stage for a lot of contemporary thinking. Okay? There's a very famous set of experiments by this group, Stanley and Miller, in the 1950s, and they dissolved a couple of rudimentary gases, rudimentary elements and molecules that were, and compounds that were known to have been around at the early start of the Earth. So it was, geoscientists will tell you there was lots of methane, there was lots of ammonia, there was lots of water. It would have been a really, really hot place. Okay, so those are the basic elements that you have. How do you get life going out of that? Well, these guys came along and they applied a spark to this mixture. Okay? And then once they did that, they could see that there was, or they claimed anyway, that they got some organic compounds that came about. Okay, so if you have this kind of biochemistry, I guess we'll just call it chemistry, it's not even biochemistry yet, and you apply a spark, there were the formation of some elements that really are what we need for self-replicating organisms. Okay? So what's cool about this is whether they're right or wrong, at least they're making recourse only to entirely natural processes. It's fully understood that there was methane in the early Earth, there was ammonia, there would have been hydrogen, there would have been very, very hot water, there probably would have been some electricity in the form of lightning bolts and so forth. So this might be the way that you get the ball game started, going from 0 to 1, and then the engine of evolution, with its two-step process, can take you from 1 all the way up to the complexity of the human eye. Right, as one of the ways of thinking about that. It's now believed that a better start would probably come from the deep sea vents that you see in the oceans. And people do the biochemistry on this. Apparently pyruvate or fool's gold is a critical element to have there in order to get this started. But that might be an answer to my early students' question, which is how do you get the ball game started? Because we seem to have a way of getting from simplicity to complexity using evolution. Okay, so questions on that? I think it's a really interesting set of slides, and again, sets the stage well for the complexity that we talk about inside of the eye, but also inside of the entire visual system. So why don't we now, finally today, get to what we were on schedule to talk about for today. We're talking about leaving the eyeball and all its complexity and finding other complexity in the lateral geniculate nucleus. We'll talk a little bit about color opponency. I wore my brightly colored yellow shirt today, the same one that I had in the video, so that we could pick up on color opponency. We'll talk a little bit about the visual cortex, how it's structured, and then the different cell types that we have. Okay? So this is what I'd like to do. Why don't we give you the chance to talk? I've been doing a lot of talking. Why don't we have you talk with each other, and maybe you can find a partner and teach them about LGN projections, these first several slides, for about 2 or 3 minutes. And then we'll have you teach me about these slides. Okay? So how do we innervate the lateral geniculate nucleus with these different axons that are coming out of the retinal ganglion cells and making their way to the thalamus? Now, why don't you find a partner, talk through those slides, teach each other, and then you can teach me.
So we're going
Okay, so I talked about loudness and nerve.
It's going to be going towards less space up here. So it is going towards the right LGN side. So all of the right side first. All right, let's see.
Okay, I realize you're midstream, but that was really cool. It's fun for me to watch people pointing in the same way that I point on the video. They're making the gestures that I was making. So I wonder if we can begin to sort of walk me through. I'll try not to project. We can go back and project the PowerPoints as we need to. We'll see how far we can go without looking at our notes. And we'll see if we can get some of this information transferred into our long-term memories. Okay. Anybody want to start us out? I'll start you out this far. So we'll pretend that I'm the person that you're trying to instruct about how vision is working at the physiological level, and we do have photoreceptors. I got that. They have different wavelength tunings. I got that. There's a principle of univariance. There's a cis-trans isomerization. So we have these photocells that are catching photons and setting up action potentials inside of the retina. Okay. Are we done? We're not done, okay? So and you're right, we're not done, because all we've done is all we've gotten so far is what's going on inside of the retina. Anybody want to start us out with what happens after that? So the retina's kind of done its thing. Now what happens? What's the next major stop? LGN's the next major stop, okay? Okay, thanks, man.
You have like a nasal side and a temporal side of each eye.
Okay.
So you have big externals like in your left visual field.
My left visual, I'm going to turn my back to you. It's not that I'm not listening. I just want to get in the same orientation, because if I face you, my left is your right, okay? So here's the stimulus out of my left visual field.
The nasal side of the left eye and the temporal side of the right eye.
Okay.
And it's going towards, they're both going to go towards the right LGN.
Okay.
So the right eye with the temporal projection side of the projection is going to be, it's the lateral projection because it's staying on the right side going to the right LGN.
Outstanding. Okay.
While on the left eye, it's the nasal, so it has to crisscross.
Okay.
The optic chiasm.
The optic chiasm, excellent.
In order to get to the right LGN.
Okay, right. So this is one of the things that we hear that was actually beautifully explained. In Intro to Psych, students learn that there's some kind of contralateral organization, and they get that much. And then because there's a contralateral organization, they sometimes say, oh, I know, it must be that my left eye feeds to my right cortex and my right eye feeds to my left cortex. And that's not entirely right. The way that is correctly laid out is what Meg just correctly stated for us. So we started out with a stimulus in our left visual field, and what is true, because light travels in straight lines, which is a fact of light, a physical fact, not a biological fact, right? It's from the physics department over in Olin, not from Talbot yet. We'll get to Talbot in a minute. That light is going to be transmitted to the right side of each eye, okay, which turns out to be, in this particular case, the nasal portion of my left eye and the temporal portion of my right eye. How many people are following that? Does that work for us? Okay. And then we do have to get all that information over into my right LGN, and she correctly laid out the ipsilateral same side projections and the contralateral projections. Okay? So it's just neat to me that it's all that orderly. right, that everything is that orderly, that we can get all the information from the left visual field to a right LGN, even though we have to take different pathways to get there. Right? They're gonna be making their way down distinct optic nerves, your left and right optic nerves. Okay? Very good, very good. Anybody, that was terrific. Anybody else want to add any details to any of that? So that was a terrific run, got us all the way to the LGN. What else might we say about ipsilateral and contralateral in the LGN? So we've got the whole left visual field represented in that right LGN. Okay, yeah, thanks.
They project out to different levels.
Different levels. How many levels do we have? We've got 6. Okay, right? All right. And we had a little mnemonic. So the ipsilateral projections land in which layers? Okay, and how are we going to remember that without looking at our notes? It makes sense because 2 plus 3 add up to 5, and that is the same as what you would expect from mathematics, and it's the same side projections is what ipsilateral means, right? Okay, so ipsilateral means same side, and 2, 3 equals 5 is the same as what you would expect from mathematics. By contrast, who can finish the by contrast statement for us? Thanks. Are contrary to what they would add up to. 1 plus 4 does not add up to 6. Okay, so the contra side doesn't add up. Okay, just a little. Maybe you have another way of remembering that. There are lots of ways to, lots of physiological tricks, anatomical tricks, but that's one that works. Okay, so now we have this nice segregation based on eye of origin, as it's sometimes labeled. Okay, if we were to leave the LGN and head over to the primary visual cortex, all the way back here, what is that road called at that point? It's no longer called Route 16 or Route 161. The same information is traveling now along a road that has a unique name. The optic tract is actually what happens after the optic chiasm and just before we get into the LGN. Okay? Then we're at the LGN and we're going to go one more. Right? We're going to go to the next relay station, or the next station, which is going to be the primary visual cortex. So optic tract was actually correct in between the optic chiasm and the LGN. Right? If we go one more, the optic radiations, okay, is going to be what we have next, optic radiations. So if you hear doctors talking about somebody who's been brain injured and there is an injury to the eye, they might say, well, you know, the optic radiations were transected. That would tell you that the injury occurred at a slightly more posterior level. So here's anterior, the front, here's posterior, the back. The optic radiations are going to be more posteriorly positioned than is the optic tract. On some level, it's, if you will, all the same road. It's not literally the same material, the same tissue, but it's carrying the same information. It's carrying the same information. Okay? Before we leave the LGN and get into all different kinds of properties of the cells in area V1, can anybody tell us about some of the properties of LGN cells? So they have some interesting properties. My shirt is a clue. Okay, thanks.
There are color opponent cells.
Okay, they're color opponent cells, right? Okay.
And there's a red-green opponent one and a blue-yellow one, and then there's the light-dark.
Light-dark, right. So we learned about light-dark color opponency, right? Or we'll call that luminance opponency. It might be fairest to call these wavelength opponents, and people who are really into physiology can tell you the maximal wavelengths that I'll call green. It's green and red, and I'll call it green and red. But they might say, well, it's actually something like, you know, the green might be 530 nanometers, and the red might be, 672 nanometers. They can actually specify the wavelengths. We'll call them green and red for our purposes here. I think it's really interesting that people who are studying genetics can look at different organisms and tell you that the blue-yellow mechanism is a lot older than the red-green mechanism. I think it's really cool that they can do that. And both of those are presumably older than the simple on-off dark light mechanism. So there are ways of figuring out how old these things are by looking at an interesting index called the molecular clock. For those people who do molecular genetics, you can estimate the rate of mutations through something like these Lenski experiments and say, well, how many generations would you need to get this or to get that? You can look across species, do some comparative analyses, and see which of our nearest neighbors have what kinds of photoreceptors and stuff like that. Right? So we have that color opponency. Okay, now we're off to LGN. We're leaving LGN and we're headed off to the primary visual cortex. We have 3 cell types there. Can we yell out what some of those cell types are? Just yell them out. Simple cells, okay, simple cells, and then complex cells, and hypercomplex cells, right? Probably poorly named, but those are the names that were given early on. And to this day, I go to visual science conferences, and I know that there are people who are electrophysiologists who still use that nomenclature. So we've retained it, simple cells. What are some of the characteristics of simple cells? What makes a simple cell a simple cell? Okay, thanks, Ronnie. They're phase sensitive. Can you say a little bit about, that sounds like a fancy word. What does that mean?
So there's like a pattern that the cell has to follow over there. There's a component like on and off.
Okay, what, do you mean when they're firing and they're not firing, or do you mean that their subfields are, okay, their subfields are arranged in such a way that the cell will begin to fire if there's a luminance increment in a certain region of its receptive field and a luminance decrement in another region of its receptive field. So this time now I think a picture will say 1,000 words. Let's call up that. And Tawny's right on track with our phase sensitivity. Let's skip over that. Okay, so this is kind of like the on and off center cells that many of you draw to in one of our TED Ed lessons. But now an interesting feature in the V1 cells is they become elongated, which allows them to pick up on different orientations. We don't have orientation tuning in the primate visual cortex prior to area V1. The cells in the retina are not tuned to different orientations, and the cells in LGN are not tuned to different orientations. All of a sudden you get the emergence of these elongated cells, but they still have the same kind of a property where there's an on-center in this particular case, an off-surround. Others have an off-center on flanks and off-center here. Okay, and then we put up a stimulus, okay, which happens to be dark, light, dark. That is going to be just right for this. If I now reposition this and just slide it on over, all of a sudden this thing will stop firing. Who's okay with that? Is that working for us? Okay. And so one of the ways to think about this is if I were to draw a simple profile of this stimulus, a luminance profile of the stimulus, I could draw a square wave profile, or I could draw a sine wave profile. I'll draw it as a sine wave, even though it's actually a square wave there. I have a region where the stimulus is very white, and then it could gradually get darker. Here it's black, here it's white, here it's black, that kind of a thing. And this is just something like a sine wave, right? And I can manipulate the phase, the relative starting position of the sine wave. So you might remember that we can say, for example, a sine wave starts here, a cosine wave starts here. Does anybody remember that sine and cosine are 90 degrees out of phase with each other? Okay. So I can basically slide this, I can grab this thing and slide it by, if you will, 90 degrees, making it half the way through, or 180 degrees. 180 degrees would be half the way through, or I can pull it all the way to where it is. Again, now I can have the dark and light right where it's positioned. Now that would be 360 degrees. So I'm basically changing the phase, how many degrees I've moved through this sine wave. And these are basically saying there's some luminance distribution out there, and it has a particular kind of profile, light in the center, dark in the flanks. Okay. How about the complex cell? That's what a simple cell does for us, the complex cell. Okay, thanks, Zach.
It's not specific to, it can have the role of responding both on and off, but it has to be alternated.
Okay, right. So there has to be a difference between the surround and the surrounds and the center. There has to be a difference, but it doesn't matter what direction that difference is. The simple cells are very picky. It has to be a particular configuration. Okay. The complex cells are less picky eaters. As long as there's some difference between the flanks and the center, they will fire. So they're telling us that there's some kind of a border out there. The simple cells are telling us which is the dark side and which is the light side. It's phase sensitive, right, in that way. Okay. The last bit that we had was, or the last kind that we had was the so-called hypercomplex cells. Okay. Anybody want to help us out with that idea? What do those report for us? Simple cells seem to be telling us about where the lights and the darks are, and we're getting luminance border results from the complex cells. Okay, thanks, Meg. They're length sensitive. Okay, Right, yeah, okay, so they're length sensitive, and it could be the case that if something extends now beyond the receptor field, the stimulus has to stop within their receptor field. And if it doesn't, then they're just not going to fire. But if it does, they might fire for us. So now we're getting some information about there's a boundary out there. Here's the light area, here's the dark area, here's where it seems to be extended. Okay, so now we're reducing the uncertainty about how the light is distributed out there in space. Okay? All right, I did want to take a couple of minutes and see if we could have somebody walk us through the tilt after effect. Okay, so I'll even do this much for us. I'll put up the tilt after effect diagram. Okay? And the reason why I like this is if we get this idea down, and we might not get all the way there today, but if we get this down, we'll explain about 4 effects later on in the semester using the same kind of logic. So we have just 2 or 3 minutes. Why don't we see if somebody can get us started on understanding the tilt after effect? Okay, I will go with Meg one more time. Thank you.
The tilt after effect is where in an experiment there's manipulation and orientation.
Okay.
And this manipulation causes an adaptation in the nervous system.
Okay.
Where we show like a reduced sensitivity.
Okay. Okay.
Use our perception physiologically, which shows it psychologically.
Okay. Right. All right. Okay. So here we have a bunch of different cells that are tuned to different orientations. This particular cell fires maximally to a perfect vertical. This particular cell fires maximally to something that is about, I'll call that 45 degrees, that's not quite, maybe 30 degrees away from perfect vertical. And that's tilted in a, what's that, clockwise direction. Okay? So you have a band of these, right? And they're picking up different kinds of orientations. Okay? And if I now have a test stimulus, well, that test stimulus is going to maximally excite this one because this is a vertical test stimulus, and it's going to get maximal responding from this cell, a little less from those, and less from here, and even less still from here. So we get this distribution of action potentials, right? For this ensemble of cells will not be firing uniformly. They're going to be firing differentially, with this one firing the most because it's got the best match to the stimulus. Who's okay with that? Okay, so this is kind of like the firing. And when this is what's firing, this is your perceptual experience. You have the perceptual experience that, hey, there must be something vertical out there. Why? Well, because predominantly it's my vertical cell that's going nuts. Okay? So you get a nice match between what you're perceiving and what's physically out there in the real world. Okay? Then, as Meg correctly noted, you can have somebody adapt or stare at something like this, okay, that's tilted that way by maybe 20 or so degrees. And after you do that for a while, those cells become diminished. They basically fatigue, is one way of thinking about that. And because they are fatigued, they're less sensitive. So now we come back and we show the same test stimulus that's perfectly vertical, but we're going to have a skew in our population response. Okay? And that skew in the population response, you can see it comparing this one to that one. Okay? We're going to now have slightly more activity on this side of vertical than on that side of vertical, and our perceptual consequence will be opposite to that of the adapting stimulus. Okay? So we're having something like an after effect. You adapt to this, you fatigue those cells, and these guys are now going to be overrepresented. And when they're overrepresented, this is the perceptual experience. So we go from the physics through the physiology to the psychological. It's a lot to think about, but we'll come back to that idea several times. How many people think they follow that? Okay? All right? Really good. Okay. I think we're just about out of time, and you've been responding really well, so I'll give you 1 minute off for good behavior. Okay? We can't wait till Wednesday. We're going to be back here having a really great conversation on power and justice issues. I'll say go, and we'll see what you've got to say. Okay? See you Wednesday.
I was just wondering for the paper if it has to be in a specific, APA.
The paper doesn't have to be in a specific APA style. A few folks have asked me about citations. If you're going to quote something, please put it in quotation marks and name the source. I don't really care if you've got the italics right in the source. Just give me some indication of where to find what you put in quotes. Okay? If you're borrowing an idea, give me some indication. I don't really care whether that citation is APA style or not. Okay?